Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And very good day to Dr. Haryati. And thank you for your time. My name is Rayhan Syahputra, and today I want to give a presentation about the method of protein purification and separation. And this topic is about the Plasmodium falciparum mitochondrial L-malate quinone oxidoreductase, or PFMQ enzyme. And this method was taken from the journal titled Biochemical Studies of Membrane-Bound Plasmodium falciparum mitochondrial L-malate quinone oxidoreductase, a potential drug target. So basically, why I choose this journal, because it was stated in the journal that PFMKO is a promising material for malarial drug candidate. And also it was also stated in the journal about the purification and separation technique of this protein from the beginning. Okay, so let me elaborate the process. The first one is about the expression system. Basically, the PFMKO codon was optimized for E. coli expression, and after the codon was optimized, the codon was inserted into PUC57 DNA plasmid at the EcoRV restriction site. And after it was inserted to the plasmid, later it was subcloned. After the codon was optimized and also it inserted to the plasmid, later it was subcloned into another expression system, which is P-Atsumo expression system. Okay, after that they will transform, the codon will transform into BL21 star chemically competent cell, which is the E. coli competent cell, for further purification. And this cell was cultured in the LB medium containing 50 micrograms per milliliters kanamycin as the antibiotics for the growth factor. And after it was cultured, we take a single colony from the LB medium and make another little culture in 5 milliliters of LB medium and 20% of glycerol stock. And after that, we make a 1 milliliter vial from the 5 milliliter LB medium, I mean the cells. And after that, the 1 milliliter vial was stored at minus 80 degrees Celsius, and it was containing E. coli strain of PFMKO or P-Atsumo. Okay, so after that, the second thing is about the overexpression in bacterial membrane purification. We take the 1 milliliter of vial and we culture or we revive the cells by using LB plate agar containing 50 micrograms per milliliter kanamycin. And after that, we take the single colony from it and we inoculate into 200 milliliter of TB medium, also containing kanamycin, and we culture it overnight at 37 degrees Celsius. And after waiting overnight, we transfer 25 milliliters from the previous culture and inoculate into a bigger size of enlameyer flask, which is 500 milliliters TB medium, and also contain kanamycin. And after that, we shake the culture with 200 RPM and also culture it overnight at 37 degrees Celsius. And this is the tricky part. We must wait we must check the culture absorbency reading at 600 nanometers, which is we must acquire a target of 0.2 until 0.5 or of the absorbency reading. And after we acquire that absorbency reading, later we add IPTG on the 500 milliliter of TB medium that containing E. coli, of course, and shake it at 200 RPM, and we culture it overnight at 25 degrees Celsius. And after that, we can harvest the culture media, I mean the bacteria. And after that, we transfer it into a smaller tube. Later, we centrifuge the culture at 3000 G for 15 minutes at 40 degrees Celsius. And after that, we can harvest the cell. And the next thing is after we harvesting the cell, we sonicate the cell, but before that we should transfer the the bacteria that previously on the falcon tube, we use falcon tube, into a stainless steel tube, because we are going to use sonicator to prevent the break of the tube that maybe can break the polymers and contaminating the bacterial culture. Okay, so after that we sonicate at 4 degrees Celsius for 6.25 minutes, and after we sonicate, we ultracentrifuge at 200,000 G for 1 hour at 4 degrees Celsius, and we can get the lysate. We collect all the lysate and add NINTA into the cell lysate. What is the function of NINTA? So basically, NINTA is a protein. The function of NINTA is to bind our desired protein, which has the nickel ion. And after that, after we add the NINTA, later we use affinity chromatography that consists of 3 types of washing. The first one is we wash using the lysis buffer. So basically, to exterminate, the first washing is to exterminate the contaminants that not bind into the NINTA, or we can call it as the resin. And the second wash, we use imidazole to exterminate the contaminant that have a weak bond with the protein. And the third wash, we use both lysis buffer and also the imidazole to wash all the contaminant that has a strong bond with the protein to prevent the contaminant. And after we do the affinity chromatography, we have the semi-purified protein and transfer it into a ultrafiltration tube, and later we centrifuge under 3000 G at 4 degrees Celsius for 1 hour. And after that, we can obtain the purified protein, and later we store in glycerol stock with 50% of volume. I mean 50% of glycerol, and also with 50% of the enzyme, and we stored it under -30 degrees Celsius. And the last thing is we determine the protein or protein determination using the SDS page, and also protein assay using and also protein determination using protein assay from Bioret. And the SDS page, before we do the SDS page, we preheat the sample of the protein for 5 minutes at 95 degrees Celsius. And the SDS page we mix the SDS page and the protein sample with Precision Plus Protein TM All Blue Standards from Bioret. And the gel staining of the SDS page are with Comesic Brilliant Blue R250 from Waco for 60 minutes. And for the gel staining, we use methanol and acetic acid. Okay, that's all from me, and thank you for your attention.